And quite fittingly, we are going to start with a presentation from Peter Kharchenko, who probably doesn't need introduction for most of you, but nonetheless, I will highlight some of the work that I find quite exciting from his group. Probably the one recent paper that he's most famous for is RNA velocity, which is this very clever idea that even if you make a destructive measurement of a single cell, you can still extract more than one time point from that destructive measurement, and you can gain some information about the history of that cell, where it's going, and what's happening with it. And he's done a lot of really excellent work in general, trying to make sense of the really, really noisy single-cell RNA sequencing data, trying to see correct patterns within the piles of noisy patterns. Now I believe he's going to tell us about integrating different data sets, which is particularly relevant to us if we want to analyze protein and RNA data together, different protein data sets together. So I'm very much looking forward to your talk, Peter.
Thank you very much. Does this mic work? Okay, great. Well, first of all, I'd like to thank the organizers for the invitation to be here, to present here. Yeah, this looks right. This is a very exciting new area, and obviously I'm learning a lot here. All right. Yeah, so we've been working a lot on single-cell analysis for the past, 5 years or so, but mostly RNA, of course. And then more recently, some epigenetic things, DNA methylation, chromatin accessibility. Obviously, we're quite aware of the fact that we're looking at transcriptional activity only, and especially when it comes down to inference of regulation and intricate sort of mechanistic details like this, the clear understanding of the protein measurements could really change the picture in this regard. And some protein measurements, obviously, we've looked at in the past. This is Tradeoff data from Gary Nolan's lab, looking at mouse hematopoiesis. And just like from my point of view, just two major things that single-cell analysis allows you to do compared to traditional methods. The first one is obviously study extremely heterogeneous tissues, right? So here, this type of hierarchy, especially in the beginning of hematopoiesis, has been studied over decades, and this is a one-shot experiment that more or less illustrates it. Now, this is purely visual, of course, but you can dive in and actually statistically analyze the transcriptional profiles if you'd like. This is probably the main application of single-cell methods today. The second one perhaps being if you're able to sort of perturb or somehow condition cells individually, then you can do single-cell readouts. This example would be perturb-seq and methods like this. Also very powerful and essentially rely on the single-cell analysis. So for the most part, we've been developing computational methods for this, and it's been kind of interesting because the progress in the field has been tremendous technologically. And whenever you talk about progress, you have to compare with Moore's law. So these are the processors I had on my desktops over the years. I just punched in the number of transistors here in the log scale, and it worked. This is more interesting. So these are sort of landmark papers in the single-cell field that just pop up in my mind. So when we kind of started doing analysis, we were looking mostly at the Azim Surani's protocols and basing our measurements on that. And there was just a handful of cells, 8, 9 cells. And then the first kind of high throughput measurement in my mind was from Stel Linnerson's lab, looking at 100 cells, mouse, ES and fibroblast cells. And already there, you could start doing some statistics. You could look at the variation properties within the measurement and so on. And it was quite noisy. And then, it scaled up quite rapidly. These are the droplet methods introduced, drop-seq and indrop. And this is not actually a publication, but the dataset 10x Genomics introduced. So as you can see, we were growing faster than Moore's law. I think it's slowing down now. I have to update the graph. But obviously, if you're working with this data, this type of growth can be both a blessing and a curse. So the data sizes are getting large, even if you're careful about keeping sort of data in a sparse format and so on. I think more fundamentally, and this is what I'll focus on today, the experimental designs have become more complicated, and now we're looking at sort of multi-sample data sets, their efforts, consortium sort of scale efforts like Human Cell Atlas to generate thousands of data sets covering different tissues. And if you want to take advantage of this, you need to be able to essentially have computational methods and statistical models that allow you to basically integrate these types of data sets. And that's most of what I'll focus on today. Now, we've gotten over the years, I think, as a community in general, pretty good about analyzing individual data sets. So if you have, let's say, a couple of replicates or a single replicate that's pretty large. So the, a typical, let's say, measurement these days would be something on that order, 10 to the 4, 10 to the 6 cells, combining different lanes perhaps. Pretty much all the protocols except for SMART-SIG2 that's still used in some cases rely on this molecular barcoding, the UMI strategy, which allows you to essentially drastically reduce the effect of amplification noise. So that basically for us, that means that we can get away with simpler statistical models. We used to fit really complex models per cell, and now we basically fit a per data set model that is much simpler than that. But on the downside, the data sets can be very shallow. So especially if you're doing, let's say, nuclei measurements, the amount of number of molecules you may get per single nucleus would be a few hundred molecules per cell, especially if these are small cells. So you can have these vast data sets that can have pretty shallow coverage. Now, it turns out that statistically, that's all right. You can more or less, in most cases, compensate by basically having more cells, compensate for shallower coverage, as long as you're able to sort of find similar groups of cells. At some point, of course, if you go too shallow, you'll cross a barrier. You won't be able to do anything with it. Now, so the tool that we've designed for analysis of such data and we've been using is Pagoda 2. Yeah, so one of the downsides of having the data growth like this is basically if your data increases by an order of magnitude every year or two, then you have to rewrite your tools, right? Because it's very difficult to write like orders of magnitude ahead. So this is the current version, and this is a data set, just public data set of 33 peripheral blood mononucleus cells. So I'll just show you some basic steps just to keep in mind the types of QC we're doing and normalization. So this is the every point here is a cell, and we're just looking at dependency between the number of molecules measured per cell and number of genes. Now, it doesn't really have to follow this very linear trend, but it turns out in vast majority of measurements it does, and it's a very quick way of basically sorting out some of the cells that seem to be poor. Now, it's a major problem, even if you're careful about identifying sort of empty, let's say, droplets or empty wells from distinguishing them from real cells. There will be some cells that are dying that are in poor shape, and they'll give you little information. There will be cells that, for one reason or another, didn't lyse well and give you little information. So you can have these types of dropouts all the time. Now, a key thing that we're preparing for next, essentially, is that these are high-dimensional data sets, right? So altogether, across all the cells, we'll be measuring, looking at, considering anyway, about 10,000 genes, let's say. So 10,000 dimensional space is very difficult to work with. A lot of things just don't work intuitively there. And now the traditional approach, is to do dimensionality reduction. But you can do dimensionality reduction in a variety of ways. The most traditional ways, such as, let's say, PCA, ICA, and so forth, they actually would like to have a distribution of the data to be nice and kind of symmetric, or normal, for instance. That would be great. Now, a key thing that we're doing, let's say, with PCA, is essentially you're trying to find a linear combination of these genes, right? In this case, that would explain most variants in the data set, right? Now, but the genes in the cell, the expression levels, they're sort of exponentially distributed. Some are extremely high, others are extremely low, and it doesn't directly correlate with sort of biological importance of it. In fact, if you look at the dependence of the variance on the log scale here with a magnitude, you will see usually these types of trends where the highest variance is naturally attributed to highest magnitude, average expressing genes. It doesn't directly follow sort of Poisson distribution. It's usually higher than that, but we could fit this type of distribution and then essentially look for genes that show variance that is higher than expected by chance or expected from this type of a model. This model is gene relative. You can have models based on controls and other things. But essentially, once you fit this type of thing, you can renormalize the variance of genes, right? So before, let's say, doing PCA, you could, naive thing would be to normalize the variance of each gene to be unity, right? But that's probably not the case. There are some genes that are more informative than others. And so we don't like sort of gene filtering. The other approach is just take the top ones and go with it. We actually would like to renormalize the variance carefully and take as many genes as possible into account, though realistically, after the first few thousand genes, you rarely get much information from there. And then we go into this type of typically clustering, in this case, KNN graph construction followed by community detection, and then some sort of visualization. So this is, I think, a large viz visualization. Anyhow, so this was actually a video that ran in real time, and after the data was loaded, it took about 20 seconds. So we actually worked hard to sort of optimize a lot of steps here because these days this is not a particularly large data set, and it's only one data set. And if you have hundreds of them, then you need to be able to do this quickly. Yeah. So another thing I wanted to point out is that in reality, even after all the statistical analysis, that simply presents the major statistical patterns in the data. It doesn't actually explain anything about what's going on here. And the interpretation of what these subpopulations are, what the processes that are happening in the cells that you measured, is more or less manual these days still. You can have some tools that can help you, like pathway enrichment, things like this. But realistically, you need to have an expert who will sit in front of this data set and browse it in a way that's intuitive and telling to them. And then we'll be able to assign labels and sort of make sense of things. And so we actually spend a lot of time writing these types of user interfaces that will present, sort of intuitive, interactive views of the data to biologists, because we need to engage them. And this is an example with like gene heat maps and these types of embeddings. So you can run differential expression here and things like this. They're costly, sort of in terms of time to code, but I think for interpreting data, it's still necessary. Hopefully, in the near future, we'll start to automate this a little bit. Okay, so today's emphasis really for today's talk is about data integration, and the motivation is really that for the RNA portion of the single-cell analysis, it's more or less mature now. So you could it's easily accessible commercially. It's not too expensive, and so people are actually starting to put it to use. Instead of sort of having one-shot demonstration experiments, they try to go after specific biological hypotheses, and that means that you have real-world experimental designs. If you're trying to address, let's say, effective perturbation, you'd have case control studies, right? Or you'd be looking at very least at biological replicates, time series, and so forth. We do a lot of sort of clinically oriented measurements, and so here you'd probably be examining multiple individuals in order to reach some kind of generalizable conclusions. Quite often you look at multiple tissues, effects of treatments, and so forth. And so the example I'll describe is our bone marrow study. So we're basically studying the effect of metastasis on the bone marrow, and so looking at many patients at different stages and with different types of treatment. And thanks to close collaboration of surgeons, we can, during surgery, actually get access to samples of different type from the same individual. So the core part of it would be, let's say, a solid tumor fraction that contains some of the bone and some of the tumor cells. So here, for each square here is a little t-SNE plot sort of independently done for a particular individual that we measured for this type of sample. But from the same, let's say, surgery, we can get a liquid aspirate of the bone marrow at the site of the disease, so matched essentially per individual. But it's a different sample. It has a different composition because you're not getting so much stromal, let's say, or so much tumor in it. We can also go into, let's say, vertebrate that at least radiographic has not been affected, and then also do a liquid bone marrow draw from there. So this is non-affected bone marrow, for instance. Again, the composition will be slightly different, and maybe the state will be different. We can compare it with controls. So patients, let's say, undergoing surgery for the reasons other than cancer. We could draw blood over time, for instance, and so forth. So you can imagine that we have this fairly vast collection. Some of it is systematically different in terms of their composition, either because of the tissue samples or the impact of the disease. And then in terms of questions, and it's quite, just quite stereotypical, so I'm giving this example, we'd like to know, what are the sort of compositional changes in these tissues that can be linked to presence of metastasis, and then also what is the impact of presence of metastasis on the state of individual cell type. Pick, let's say, a particular CD4 cell, T cell, and then subtype, and then understand, how it's being impacted. Of course, we'd like to analyze the effect of treatment as well, but that, in all fairness, will require more samples. So what we would like to do is to, for pretty much all of these types of downstream analysis, we would like to have a way of jointly analyzing this entire collection. So at the very least, we would like to be able to track a particular cell type or subset of cells throughout the collection if they exist there. Right, so instead of having these analyzed independently, we basically want this kind of a uniform coloring where if you see, let's say, cluster 17 here and cluster 17 here, it indicates that it's the same state, the same subpopulation, more or less. And that's essentially what we've developed a new method for. Now, a couple of related problems to this, and I just wanted to kind of separate them out a little bit. So the traditional one is that of batch correction. So typically if you have multiple replicates, especially technical ones that you've done, there will be some variation between them. You want to apply batch correction. Now the key assumption there is that whatever difference you're seeing between the samples is essentially artifactual, and you need to explicitly get rid of it, right? So you're correcting for technical defect for that matter. Now, a related problem is that of sample alignment. So if you have a pair of samples that are similar in composition, let's say biological replicates, and they will be a little bit different because biology is different, and you'd like to put them together. So there are a couple of papers that came out last year from John Marioni's group and Raul Satij's group that aim to do this. One is based on canonical correlation analysis, and the other one is based on this mutual nearest neighbor mapping. Now, all these methods still basically aim to bring this data into sort of a common expression space, right, or same space. And when faced with these kind of large heterogeneous collections, we thought that it's actually making problem more complicated than it needs to be. Essentially, for joint analysis across collections, especially very diverse collections, we don't need a common space. In fact, it's hard to imagine in certain cases, let's say you're integrating between species. But what we need is a way of mapping from one data set to another to basically establish this type of correspondence. So that's the general idea. We're going to try to take the collection and just wire it together in a way where we can statistically test for associations between different groups of cells. All right, so the approach works as follows. The first part is quite similar to the sample alignment. Essentially, we take a pair of samples in this case, and we'll come up with some kind of a rotation to basically look at them in the right way. Remember, we need to basically move away from the high-dimensional space for most analysis to be effective here. So we tried a variety of rotations, and I'll try to illustrate them. It doesn't actually matter that much, but the consensus principal component analysis seems to be one of the more powerful ones, and a simple trick of doing independent, basically combining independent eigenvectors from the two data sets seems to work just as well. So, after we do this type of rotation, we can establish sort of a putative mapping between the two data sets, and essentially we'll assume that it's error prone, but we try to do the best we can. So the simple strategies are basically finding the nearest cell in the other data set or the best reciprocal hit, so the mutualist nearest neighbor. And so, for instance, these red cells may map to these red cells or and so forth, right? Now, the key thing here is that we'll continue doing this between all pairs of samples. So here, for instance, we have only 3 samples, maybe others. And if, let's say, this red population actually represents a recurrent population in the collection, and then these red cells will tend to map to each other across these multiple pairwise comparisons, right? And if you imagine you have a collection, let's say, of 10 samples, you're doing quite a few pairwise comparisons. And so these red cells will start to form this type of a clique. And altogether, we have essentially a joint graph here. And then these types of cliques are easily recognizable, statistically within the joint graph and by a variety of methods. We do also maintain within a given data set some edges in the graph that connect cells that were nearby within a single data set. But these are pretty weak edges, about 10 times weaker. So essentially, they represent sort of a weak prior for clusters that have been observed in a particular data set. And the reason why we keep them, if you imagine that this blue subpopulation existed only in this one sample C and no other sample, then in order for these cells to basically stay together, we need some sort of a weak prior in them being nearby. Now, once we have this joint graph constructed, we can use it for a variety of downstream analysis, such as visualization, simply laying it out, and then clustering, as I'll show you, and propagating some information. First example. So this is a data set from Human Cell Atlas that was prepared at the Broad by Chloe Villani of 8 bone marrow samples, human, and 8 cord blood samples. So this is the joint graph, and essentially already clustered and annotated. This is just the composition of samples on this joint graph. Now, you'll see some systematic color difference, but basically these tissues, while they share a lot of cell types, they are systematically distinct. So let's say coverage of progenitors is a lot lower in the cord blood than in the bone marrow, as one would expect. Now, if you look back at sort of the individual, let's say, samples, and here just the individual t-SNP plots for each sample, colored using basically the same joint clusters, right? So then you can go in and see that this pink cluster would correspond to this pink cluster and so forth, right? So it basically gives you an initial way of starting to do joint analysis. Now, we try to compare it with sort of existing methods, and just conceptually, I wanted to emphasize the types of properties we're looking for. So the key thing here we wanted is to have a robustness, right, of the response. So if you, let's say, detect these subpopulations, if the problem becomes harder, or if we're pretty sure of these populations, but if we make the problem a little bit more difficult, that we're still able to recover them. And the first one is pretty easy. We're just going to downsample the cells randomly, right? So we'll have less and less coverage. You want to make sure we're still recovering these joint populations. And the other one is a bit more interesting. So we designed this method to basically handle very heterogeneous data sets, right, or collections. So what we'll do here is we'll go into each sample and randomly drop one of the clusters in a sample, and go into the next one, randomly drop some other cluster. So with the probability P cluster emit. So basically we're increasingly making this collection more and more heterogeneous and samples less similar to each other. And again, the property we're looking for is ability to sort of still recognize these joint populations as collection becomes more heterogeneous. Yeah, I mentioned that the actual, let's say, neighbor mapping or the rotation that we use doesn't matter as much. So here, for instance, this graph was constructed using mutualist neighbor mapping, and this is constructed using nearest neighbor mapping. And pretty much, the results are very similar. Now, the reason is that because the key power of this approach actually comes from these multiple pairwise comparisons. So essentially, you take, in a large collection, you take a single sample, and you reflect it more or less on every other sample that you have. And that gives you quite a few comparisons, and that allows you to correct for sorts of random variation that happens between the samples. Another kind of property we're looking for is basically increase in statistical power as the collection grows. So imagine that you were sampling just the blood, normal peripheral blood monocytes of different individuals. Now, each one of us is actually quite different, right, compared to, let's say, mice. Mice will lay on top, the data will kind of go on top of each other all the time. But with humans, there's variation. So on one hand, you're increasing the collection size, but you also have this background variability between individuals. So the property we're looking for is that as the collection size gets larger, we're able to distinguish statistically recurrent subpopulations that are more and more subtle. Let's say finer subsets of CD4 cells and so forth. And we don't have very large collection here, so we're only going up to 10. Actually the much larger collection is coming, and we can test this, but at least, and this is a number of stable clusters we're detecting, and so at least the asymptotic relationship is in the direction that we would like. And this is just kind of following up on this bone marrow study, the example of how we use it. So this is the collection that we have from different fractions that were collected during surgeries and controls. And essentially, after you join them in, you have this view of more or less uninterrupted hematopoiesis here. And then these are the tumor cells nearby here. T cells mature elsewhere, so they're detached essentially here. Now, once you have this kind of integrated view and integrated clustering, you can start doing all kinds of analysis. So for instance, here just looking at compositional shift between benign controls and the cancer controls. Have, let's say, benign controls much higher B cell generation. You could start looking at transcriptional shifts within populations and so forth. All right, so I'll go over and show some examples sort of increasing complexity down to sort of almost ridiculous ones. So first, a simple one. This is a realistic data set from head and neck tumor published in Cell a couple of years ago. This is using SMART-SIG2 measurements. And then, as you can see, this is actually quite typical of if you're working with primary tissue still, a lot of it is quite challenging to process, right? A lot of samples will yield relatively few cells. The cells will be in poor shape, and then the survival of the cells is selective, right? So you might end up having more of one cell type versus another. So in some of the samples, you'll basically get something like this, poorly informative measurements that more or less maintain a single cell type. While others will give you a nice kind of map of subpopulations that exist there. That's just reality. So, so these are the joint graphs for this, and basically I'm just showing annotations that were presented in the paper, and these are the joint clusters determined by a method. And this is the composition of the samples within this space. Now, more or less, all the samples are represented in all major clusters, but you'll see that there are some clear overrepresentations. But let's say T cells, there are a couple of samples that are just giving you the vast majority of T cells. That's just how the composition of the tissue worked out. But overall, the mixing is quite good. Now, a conceptual point I'll try to illustrate based on this breast cancer data set is that there's this kind of, whenever you're doing integration, there's always going to be a trade-off between sort of the resolution and the breadth or the generality of the integration. So here we have samples of coming from multiple tissues, blood, lymph node, and tumor in these breast cancer patients. And this is, again, the joint graph. And this is showing just the tissue composition here. Now, if you think about cell types, that generally can be thought of as hierarchical clustering. You have, let's say, immune cells, you have T cells, you have CD4, CD8 T cells, and so forth, right? Now, not everything is hierarchical. Obviously, if you have, let's say, immune activation or cell cycle, that's going to cut across the hierarchy. But as far as cell types are concerned, you can think of it as hierarchical. So this is hierarchical structure for this. And the only thing I did here is that with thickness, I'm representing the entropy of this branch. And the entropy essentially rescaled to show how many samples are contributing to that particular branch. So if it's thick, that means that it captures a broad set of samples. If it's thin, it's just a single sample. Now, also, we had multiple tissues here. So I'll just show the composition of the tissues with this color code. And so if you have a node like this, that's more or less balanced between the tissues. And this, for instance, is primarily blood. So this hierarchical tree, essentially, we can cut it at different levels, right? So if we cut it here, let's say, where we have a nice mixing of different tissues. So we're getting sort of a general cluster on one hand, right? That is present in all the tissues and can be used as a unit of analysis across the tissues. On the other hand, all it gives us is the separation between CD8 and CD4. So the resolution is really poor. We're not really getting at sort of fine details here. If you cut it lower, for instance, yes, you're getting a lot of subtypes, but a lot of these subtypes are already specific to the blood, let's say, to lymph node, and so forth. Now, this is sort of expected because if you fish out, let's say, CD4 cells from the blood and from the lymph node, they will be systematically different, just from biology. There will be systematic differences that distinguish them. Same thing would be perhaps for disease and control sets. So you expect them to cluster together a little bit nearby, right? And that's essentially what happens. So in doing this type of integration, unless you explicitly force this procedure to control for a particular factor, you typically get these types of results where you could have a broad interpretation that will give you worse resolution, or very detailed interpretation that will not integrate as evenly across the entire collection that you have if it's heterogeneous. Now, because it's not really a mathematical problem, it depends on the question you're asking, we basically made a UI where a person can navigate and explore this and pick a cut that they like. Now, a little bit more on that topic, but slightly different technical angle. So here we have data sets that generate using different platforms from pancreas isolates. And so we have, integration works pretty nicely. You have, let's say, beta cells, alpha cells. And if you look at the distribution of these platforms, I think there's like 4 or 5 data sets per platform, right? But here I'm just showing platform coloring. So if you look at the distribution, you'll see that, let's say, in the beta cells, while they're all kind of clustered together, but there's still a separation of the, say, 10x measurements and in-drop measurements. They're nearby, but they're separate, now sort of separating. Again, because these different platforms have their own biases, each one of them, these cells, the state that they're reported for these cells will be systematically different, and at some point, the methods will pick up the difference, they'll start separating them. right? But in this case, we know it's a technical difference, right? So it's fair for the user to say, okay, I know that these were measured on different platforms. Here's a factor describing that design. Can you please control for that? So we can do a few things to basically force the alignment to be better, and I'll just describe. One general strategy is to sort of defocus, right? So typically when we, let's say, compare a pair of samples in this elementary procedure, we'll look for the neighborhood, let's say, over here, the closest cells, let's say, to this cell over here, and then we'll map back, and then if let's say, any of these cells find as one of their closest neighbors this cell again, then we'll draw a link, right? This is the best reciprocal hit link. Now, what we'll do instead is when we're mapping forward, essentially, we'll look at much larger neighborhood, but we'll only sample a handful of edges from it, right? So essentially, we on purpose sort of losing our focus and looking at the broader context there to pick up more connections. And so we call this alignment strength. So it's just a number that you can start to increase. And here I'm increasing it gradually, and gradually the mixing becomes a little bit better. Now again, just to illustrate the conceptual point, if I keep increasing it, it will become fuzzy and fuzzy, and then if I set it to maximum, it will be this. So just to point out, what happened here is that I've asked the algorithm to explicitly generate the perfect mixing, right, between platforms. And the only way to ensure perfect mixing is to basically lose all the structure of the data, right? So again, just to illustrate this kind of a balance that we constantly have. All right. So the other thing you could use a graph for is to diffuse information about discrete and continuous labels. So discrete labels would be, let's say, actual labels, let's say annotations for the data. If you have, let's say, a data set of your favorite tissue that an expert actually annotated in the lab, and then you generated 5 more, you don't want to call that expert again. You could probably figure this out computationally. And so if this one data set is annotated, what we can do is take the labels from this data set and then simulate a diffusion process through this graph, and then see what labels arrive to different cells. That's essentially the process. So here, I'm going to practice with this bone marrow data set, and what we'll do is we'll forget the labels for everything but this one data set, and then we'll try to propagate it back to the other 7 and see whether we got the same label or not as expected. And these marks the places where we guessed it incorrectly. And for the most part, the mistakes are at the boundaries of the clusters. That's sort of expected because if the cell is at the boundary, it means likely its membership is unclear. But also, we can monitor, as this diffusion simulation takes place, we can monitor what types of labels arrive at a different cell and estimate the uncertainty in a label. So this is the reported uncertainty in a label. And essentially what this shows, same regions, it shows that we made a mistake here, but we basically knew that we were likely to make a mistake at these cells. Now, the other thing you could do is propagate, let's say, continuous values. A good example of this is propagating, let's say, expression values themselves. So if you propagate the gene expression values themselves, you essentially smooth the expression across the data sets. It's the way we use, for instance, to arrive at this common expression space if we need to, although we don't recommend using it in general. If in one data set you measured something else, let's say you measured accessibility and the other one you measured RNA, you could use this type of propagation procedure to basically do imputation. All right, so more extreme examples. So here we're looking at an atlas-like collection starting with tabular mirrors from Chang Zuckerberg Foundation. It's published last year. So basically they use 2 different techniques, 10x and SmartSeq2, to look at many different tissues in a mouse. In a kind of great collective effort at the BioHub. And this is the joint graph just with clusters. Altogether, it's 46 data sets, about 100,000 cells. And this is the distribution of platforms of these 2 platforms that they had. Now, there are places where they mix, but there are places where they clearly don't mix. And if you look at the strategy that they had for sampling, or how they ended up sampling anyway, you will see that a lot of tissues were sampled, let's say, with one platform, but not another. So we don't expect a good overlap there. And this is just the higher, magnified view of this joint graph with the labels that were published in the paper. Now, I'm not going to focus on this so far, but I'll show you sort of the next iteration. So here, for instance, we take this tabular mirrors data set, and there's also, at the same time, more or less, was published a an atlas, another mouse atlas from Geiji Guo's lab using microwell technique. So that gives you much shallower measurements in terms of coverage, but more cells, in this case 300,000 cells, about 81 data sets. So here altogether we again feed these 127 data sets into this integration, and this is distribution of platforms again. You'll see that more or less the same trend. There's some clear overlaps, but there are plenty of regions where there's almost no overlap. So even though these two teams did an amazing job, I think, sort of pioneering job doing these types of whole organism atlases and spent a lot of effort, from the sort of lack of overlap in a lot of these tissues, you see that we're pretty far from having sort of a comprehensive, a well-covered mouse atlas anyway. This is right. So this is again the view of tabula Murris annotations. Now it's pretty messy, and this is just for Bogdan. I know he loves T. This is not actually a T-SNE, it's called large viz, but it's same idea. You kind of have to look at it as abstract art. But for instance, here we have hematopoiesis, and it's more or less laid out, where you have progenitor cells over here, T cells, and this T branch, in this side B cells, and then neutrophil, macrophage, and let's say erythroid, I think, goes up, and so on. So it laid it out reasonably well. But of course, I'm not insisting that this type of view of an organism is somehow most informative one. All we're looking for is just a way of quickly visualizing it and integrating it. Now, most of the analysis, obviously, you basically take certain subpopulations that you're interested in once you're able to find where they are, and then focus on them with sort of downstream analysis, say differential expression, things like this. All right, more interesting things, different modalities. So here we just take attack-seq atlas, so chromatin accessibility measured in Jay Shendur's lab. So it's 17 data sets covering 13 tissues, and here I'm showing the same joint graph. Here are the data sets on that joint graph, and here are the annotations that were published there. So it's just showing that we could do the same kind of procedure on a different modality, in this case attack-seq. Now, more interesting is integrating across modalities, because we can actually learn something new there. So here for the long data sets, we take this accessibility data from Jay's lab, and then from these mouse atlases take the RNA data. And again, showing the same joint graph, but here I'm showing only the cells that were measured for RNA, only cells that were measured for accessibility, and this is just the distribution of the platform. So we have a couple of attack-seq data sets, and then 3 different RNA-seq platforms, right? Now, what you can see is that, and then the annotations are the ones that were published in the corresponding papers. So you can see that even though the naming conventions are very different, but it maps more or less the same cells, stromal endothelial cells here. T cells, for instance, separating NK cells and T cells, so even resolution is not bad. But if you look at this type of a platform separation here, you'll see, for instance, for this stromal group, the RNA data sets are grouping closer to each other than the 2 attack-seq data sets, and they're next to them but a little bit apart. Again, because no matter how we try, the accessibility data will be systematically different. Now, what I didn't tell you is actually how we did this, right? Because in order to do this kind of pairwise comparison, you have to have the same kind of set of variables, right, to compare. So it all depends on the kind of a linking measure that you use in this case. Essentially, we'll try to take, in this case, we'll try to take accessibility and make it transform it into a variable that looks more like expression, right? We could even come up with a third latent variable and transform both expression and accessibility to that variable, but you need some kind of a common variable in this case. So in this case, I used gene activity scores from Cicero, which is algorithm from Cole Trapnell's lab. But a simpler way to think about and calculate this, for instance, is to take a region around promoter proximal site, just take average accessibility around promoter, right? That correlates okay with the gene expression, and that kind of low-level correlation is enough for you to start establishing these kinds of connections. But if you make the problem harder, let's say you try to shrink that window around a promoter down to, let's say, a 1 kb. Now, at a certain point, you'll lose that signal. It will become non-informative, and then the integration will break down. And essentially, these cells, for instance, will fly apart. They'll look like separate clusters. Now, of course, so first of all, the linking measure is critical. And the second, it's hard to know whether, for instance, if you're integrating across modalities, is a fault and is a problem in your linking measure, or actually you're looking at different cell types that can be easily joined. It's hard to tell. And I think a lot more work will be needed in this direction. All right, so hot off the press here, wanted to thank Nikolai for actually putting both the data and the preprint, and this is the data you've seen yesterday, and so can we try to integrate this? So this is just my reanalysis of that data, and I started with a normalized matrix there, and then looked a little bit at the raw matrix as well. So again, this is just combined data matrix as is, and if we do this type of annotation using this, for instance, Pagoda 2 software, just feeding in these values, you get a nice separation between the monocytes and macrophages that are in the mixture. These are sort of unbiased clusters determined, but you see strong batch separation. For instance, there are 2 batches in here, and obviously that's picked up. That third population is a bit interesting, and we'll see it pop up again. So the first thing we can do is basically feed these data sets independently into this integration method. And so then if you do that, then you essentially correct for batch effect. So now if you look at the batch, it's evenly mixed. And you get sort of nice separation between the macrophages and monocytes. This cluster 3 still pops out on the side a little bit as a subpopulation. All right, so that's the protein side. Now, RNA side. So there, I think the issue is that, so this was done in vitro, right? We have quite a few in vivo data sets with monocytes, macrophages, but macrophages in particular are very diverse. So I'm not sure I'm picking the right data set here, but that was my best bet. So this is from lungs, and it's a particular patient in lungs, and it shows a nice kind of gradient, both monocytes and macrophages in this case, plus some other cells that are unrelated to this analysis. So we'll just focus on this a subset here, and this is CD14 monocytes, and this is a macrophage portion. Now, going back a little bit to yesterday's conversation about sort of coverage, occurrence, and so forth. So this is, for instance, looking at RNA, and here I'm showing on the log scale the number of cells in which a particular gene has been observed and the number of such genes. So overall, you kind of have this distribution, a lot of genes close to 0, and these are highly expressed ones. And the blue portion here shows basically whether these genes were observed in the protein measurement or not. So as you heard yesterday, basically the protein measure is really focused on the more highly expressed ones. So you see this kind of expected bias here towards the end. But you also see this, that is detection of proteins that on RNA level we don't see a lot, right? And that's great. It means we're getting additional information there. Now that's the flip side of it. If we just look at the proteins and just look at the fraction that is in RNA or not in RNA. So most of them are detected with reasonable threshold, but there's some fraction more or less evenly distributed across the range that is not detected in this case. But overall, the overlap is not bad. We could try to do integration. So if we try to do integration, we get something like this. So the RMAC, these are RNA-determined macrophages. PMAC are the protein-determined macrophages, right? So these are kind of the annotations that we had in the beginning, and that's how they fall together. So you have this. This is the RNA monocyte macrophages. The protein macrophages sit somewhere in between, closer to the macrophages, and the monocytes from protein are all the way here towards the extreme end of the monocytes. This is showing the data sets, right? So the green and red are the 2 protein data sets, and these are the key markers here. So it's not bad. Now, if we actually look at the RNA clusters, so this is the original RNA data set, and as you remember, it had a bunch of clusters here. So you'll see that it's actually not a random distribution. So this big cluster 9 sits on the top, and it's really above the protein macrophages. And these are, I think, alveolar type macrophages. I think it's just unique to the lung, and we're not seeing them here. Nevertheless, they are macrophages. There's this region that is actually reasonably well integrated between protein and RNA. So these are the closest matches. And then the close matching monocytes are somewhere here down at the bottom, corresponding to this cluster 2. If you look at the composition, so these are joint clusters determined on this integrated data set. And then the composition, the protein essentially cells, for the most part, fall into cluster 2 here, and then the cluster 4 for the monocytes, right? That's where the composition differs. Now, it integrated reasonably well, but of course, what I was interested in is sort of the strength of a signal and the extent to which we can do these types of linking. So here, I'll just try to illustrate visually the consistency of differential genes, in this case differential expression. So here we're looking at differentially expressed genes between monocytes and macrophages determined based on RNA, right? So the yellow are macrophages, the blue are monocytes here, and the rows are different genes, and the columns are different cells, right? So this is the differential expressed genes determined from RNA, shown in RNA. And you see this kind of diagonal pattern, or diagonal pattern. Now this is the same genes shown in protein cells. And visually you could tell, especially in the monocytes, that they're differentially expressed as well. Now this is doing a flip side. So here we determine differentially expressed proteins, basically, using the protein cells. And you see the nice kind of layout here. And then look back in RNA. Not so nice. So you could see some downregulation here, but generally speaking, things are a lot more washed out. Now, there's a critical parameter here that actually has to do with this detection and thresholds and so forth. So in determining protein D genes that exist in RNA, I have to basically tell how well they were measured with RNA. So right now I put like a bare minimum threshold. They have to show up in like 10 cells. If you make that higher, and then the subset of consistent genes will actually be a lot better. But nevertheless, it seems like if you're selecting differentially expressed proteins, then the consistency of RNA is worse than doing the other way around. All right, so quick summary. So basically this Conos software, and by the way, all this stuff is open source, and it's up on GitHub and should be published soon. There's a preprint on Barchive too. So it's basically an approach to wire together these large single-cell collections. The key idea is that we're going to do many pairwise comparisons, reflecting each data set on each other, establishing these types of putative mappings, but we expect them to be error-prone. And nevertheless, because we have many comparisons, statistically within the structure of the graph, these types of subpopulations and cliques will be quite apparent. And so we can use this joint graph to basically do, say, joint clustering, information propagation, and all kinds of downstream analysis of the data. The key thing we're trying to handle here is variability of the samples, the heterogeneity of the collection that we're doing, and make sure that sort of our power to detect things grows with a number of samples. And as far as integration across modalities, I try to emphasize the importance of these linking measures that I think need to be modeled and derived a lot more carefully. And finally, wanted to thank people who've been involved in this work. So Nikolai Barkas has worked on this initially, and then Viktor Petukhov has worked on it after that. So he's here, as well as my PI collaborators, Konstantin Khodisevich, David Skadden. And this is the group with Phil Saylor, who's working on bone marrow metastasis. Thank you very much, and I'm happy to answer questions.
Thank you, Peter. That was a real treat, especially the last part for me. I loved it.
Well,
this is exactly what I would love to see happening. Data being shared, being reanalyzed by the entire community. You could not have epitomized this any way better. Thank you. Questions for Peter? Bogdan has a question over there.
Yeah, it's a great talk, really great talk, and yeah, a lot of pictures as I like, and I really love, protein pictures for the first time. It's really great too. My question goes back. Could you just go to the your two plots with the RNA-seq and proteins?
Yes.
Yeah, this one.
Yeah.
Does this means, in your opinion, then if you go to the 2 second squares, right, go back to the RNA-seq, which we don't see the good separation.
Here. Yeah.
And all those RNA-seq been used in, for example, the previous data set where you took them from, right, from the RNA-seq.
Yeah.
I'm not sure how big this list is. Is it 1,500 lines?
It's 200, 100 on each side.
Yeah, for example, 200. Does this mean actually that if any of those 200s in original set been used for make a grouping in the original RNA-seq paper, that means has nothing to do with the proteins. It's just picture being drawn by RNA.
Yeah, that depends. So RNA data is remarkably robust in most cases. So
no. My question is not about robustness. My question is about the pictures being drawn by this data.
Yeah.
Has nothing to do with the final product, with the protein.
Yeah, so if you go back, let's say, to this initial picture where I introduced the data set, so here I analyzed the RNA data without using any protein, right? And these genes were used there because we use a very large set of genes, but they're renormalized according to this relative variance. So maybe not all, but probably most of them were used here because they're still differentially expressed, even though weakly. But like I said, the RNA signal is quite robust, so whether we use them or not probably won't make any difference. In fact, we often do this exercise where we determine top differentially expressed genes, let's say for these two clusters, let's say 100 genes, and remove them and reanalyze it. And 2 clusters pop up again, and we remove them again. So you can keep doing this surprisingly a lot, because the actual sort of the expression manifold that we're looking at is very simple, right? And so many genes support it. So I think, picking on individual genes doesn't help in this case. What I was trying to illustrate is just general kind of correspondence between the two modalities.
Yeah, which looks pretty poor, actually.
Well, I was pleasantly surprised, honestly.
The first two I like it. I mean, it's the reverse things.
Yeah.
That means those 200 on the left bottom square have no information basically about how actually proteins looks like.
Right. Well, that's true. So one thing to keep in mind here, and I try to emphasize, is that it's kind of hard to calibrate this because the protein measurement by design is focused on the highly abundant proteins, right? With RNA, this is kind of a threshold that I set personally, and if I set it to be more stringent and then look for overlapping genes, this picture will improve, right? Here I set it to be reasonable but not stringent. 100. I think. 10 cells or so. But
I think we are not yet in a position to really determine how much post-transcriptional regulation there is from these data. At the very least, we should sequence exactly the same single cells as Peter suggested. I fully agree with his comment. Or, even better, we can sequence the RNA from the same cell and measure the protein and then do the analysis. And we're going to hear more about that in a little bit, actually. But I think that's wonderful first foray. It's just the very beginning. It's too early to have a conclusion.
Yeah, I would not take some kind of a conceptual message from this. This is purely technical. If you're trying to align the data sets, how well does a signal match up? The reasons why it does or does not match up can be more complicated. I think that we need to think more about it.
No, it's a great data. First 2 squares clearly show that there is a beautiful correlation there.
So central dogma holds.
I think it looks amazing.
I have 2 questions. One with regard to this. Did you also look at the differences between protein and RNA patterns? You are looking what is common between them. If you look at the orthogonal space, what is not common,
you could certainly focus on...
You could see something interesting.
Yeah, I didn't do that. This is in some ways unbiased, right? So if you determine, let's say, differentially expressed proteins and look at RNA, if they were clearly different, and that's kind of what we see. But I understand your point. You could explicitly look for things that are distinct, let's say different axes of variation. I'm sure they exist. I didn't look for it, and honestly, I think going forward with more uniform designs, it will be more appropriate. Because here I'll probably find things that are specific to lung versus in vitro.
Okay, and the second question, when you have this graph type approach that you compare different samples without doing the normalization one to each other, if 2 samples have a very small difference between them, let's say the 2 red populations of 2 different patients, only 1 gene is different between them. What happens then? After you do your procedure, can you still see it?
So the key thing is that, and I think that's what I meant by not taking them to the same expression space. So first of all, the wiring procedure, the construction of the graph, and the determination of these subpopulations is quite robust to these types of things. You could test it by throwing out genes, by doing different normalizations, like one is log scale, one is linear scale. It still works. So it's designed to be robust. However, all it gives you is this kind of mapping, right? So in the downstream analysis, let's say if I'm interested in the differential expression between my disease and healthy controls, right, for, let's say, T helper cells, I'm not going to use any kind of smoothed expression values. I'll go back to the raw expression values, consider the design parameters like platforms and so on, and reanalyze differential expression, starting again from the raw expression values. The only thing this would give me is the set of cells that should be compared to each other. Right, but I think whatever, depending on your downstream analysis, the statistical considerations that you need to take into account can be very different. So I don't think it's possible to come up with a sort of imputed or uniform matrix, that's corrected matrix that you can do downstream analysis on that would be universally applicable. I think that only works for visualization, essentially. So we always try to, go back to the raw data and then reconsider, what types of corrections are needed. And this, in this case, is primarily used to just establish, which subpopulations should be compared.
It's great work. Definitely like better linking measurements will enable better integration. But on the flip side, do you think integration can also inform regulatory mechanisms among different modalities?
Absolutely, right? So the reason why we would like to be able to look at, let's say, epigenetics, like chromatin accessibility and expression and protein, ideally, right, is to be able to infer the entire sequence of, transcription, expression, binding, and upregulation of, let's say, of downstream factors and so forth. Now, ideally, we would measure it all in the same cell, but, if we're not able to do this, but we're able to get, sufficient number of such cells, then this is the approach to do it. We can try to integrate it, and we would have some cells that give us RNA, some cells give us epigenetics, some cells will give us protein, and then you can try to do this type of analysis. So this is, one obvious sort of example, but I'm sure there are more intricate ways of using it.
Hello, here, questions. How do you do the differential expression analysis for the proteomics data? Because currently, most of the differential expression analysis tools for RNA sequencing data is for continuous data set. So if applied to proteomics data, should have some restrictions. So how do you modify or adjust?
Right. So I went with a very naive thing. So first of all, so what I use is just Wilkinson rank test in this case. So it's a non-parametric test. I used it. The only caveat, I used it on the imputed data that was deposited. Now, probably more proper would be to use it on the raw data, taking NAs into account. But we discussed yesterday, there's some tricks with that. Now, the comment to that is actually for RNA, we pretty much switched to non-parametric tests as well. And the reason is was mentioned yesterday is that if you're comparing, let's say we started with like comparing 8 cells and 8 cells, I think, in our early study, and it was very critical to squeeze out as much statistical power from that as possible to identify the genes that were differentially expressed. So we built these complex models and parametric tests that took into account. But if you're comparing 500 cells and 500 cells, it doesn't matter. The statistical power is incredible, right? And you can use non-parametric methods that will have less bias and less problems and are very well developed and fast. So we basically changed to that. I think in the differential expression realm, as you can reconsider this problem for the proteins, you may need to take some statistical models into account, but eventually I expect you will get more or less there. And then you can either, if you're doing a location test, that will be a situation. If you're doing, distribution shift test or like presence-absence test, it might be slightly different, more complex statistics that's needed. But for location tests, I don't see a reason, to be honest, to do anything fancy anymore. Even though our first paper was differential expression analysis. But all right.
I have a lot of questions for you, but I'm going to follow up with that afterwards. Okay. So let's thank Peter again.